on mutagenesis. So start with a very basic question. How many genes are in a genome? And if you look at the abstract from the soybean genome paper and zoom in, bioinformatically they predict many, many genes. Again, bioinformatically, and the simple question is, well, what do they do? And even more so, 70% more than Arabidopsis, so unique genes that we can't even get from the workhorse of Arabidopsis genetics. And so we're looking to do mutagenesis work in the soybean to really add to the phenotypic diversity available. So we're moving outside of Arabidopsis. And so we've already heard 2 talks today about what we can do with mutagenesis and the sort of phenotypic variation you can create. So we already heard about EMS, but there's also fast neutrons, if you're familiar with that. They're very useful for plant species with large genomes. They create large structural rearrangements, big deletions, knocking out many genes all at once. And then we just heard a great talk on CRISPR-Cas9 for precise genome modifications. Today, an alternative approach using transposons. And actually, not even a dicot transposon. We'll be using a monocot transposon in a dicot. So why care about mutagenesis, especially in soybean? Well, it increases genetic variation where there might not be any, which will allow you to confirm the function of genes that you're interested in. And so this is an example of one trait we were able to create that didn't previously exist. So brown-seeded soybeans, that was caused by a transposon insertion. And then ultimately, for people who are interested on the evolutionary side, this is creating greater functional understanding of genes, that if you come across a candidate, maybe soybean will be closer to your species and you can get greater insights. So it is a resource both on the agricultural side and the basic side. And so we're using a transposon called MPING. It comes from rice. It's very short, so it's a mite. It's a DNA transposon. It's also a deletion derivative. So it was a part of a much larger transposon called PING. So MPING stands for miniature ping. So it's lacking all of this information, including these 2 open reading frames that encode the genes necessary for transposition. So MPING by itself can't move, but if you transform it with these 2 genes, it can jump and mutagenize the genome. So about 8 years ago, it was transformed into soybean, and since then it's created a number of traits all across the life cycle of the plant: leaf shape, pod color, seed color. Plant architecture is really big if you're growing soybeans, just how densely can you grow them. So it's active and it's creating phenotypes. But genomically, it's got some interesting properties as well. So it does tend to land near genes, so you don't need to really go through all of this bar chart, but the notable part here is that if you look just upstream of genes, the black bar being the observed amount of insertions, it's greater than you'd expect by chance. So it's kind of interesting, not landing in genes, but just upstream as well, just downstream. So if you're looking for a transposon to mutagenize a genome, it's a good quality that it's at least landing in the neighborhood of genes. And then secondly, each triangle here is a transposon insertion. These are the 20 chromosomes of soybean. It tends to land in the arms of chromosomes where the genes are located. So another attractive property of this transposon. And so it's been transformed into soybean, and now we're really trying to monitor its location. So it's moving around. We need to know where it's landing. If it's in an interesting gene, we can follow up on that gene. And so to get there, we use high-throughput sequencing, and it's a very simple library protocol that if you're doing genomic work, you're probably familiar with the major steps. Fragmenting your DNA. Here the red rectangles are transposon, ligating on adapters, and then this is the one difference from the library preps you're probably all doing, is using an outward-facing primer in the transposon to enrich your libraries for transposon-adjacent DNA. And then we add barcodes, multiplex these, sequence it using Illumina technology, and then map it to the reference genome. And then once we get that data, we confirm those insertions with just simple PCR strategy, using one primer anchored within the transposon and another one next door in the genome where we think that the transposon landed. And you really do have to do these conformational PCRs because you get a lot of false positives. So my undergrads they love MPING, but sometimes they get really disappointed, like, oh, this one wasn't real. So it's really important to do these confirmation experiments. And so we also use tissue culture. So we just heard a little bit about tissue culture. We sometimes use tissue culture to increase the rate that transposons jump. And so for this particular experiment, I'll walk you through this gel. B stands for before tissue culture, A is after, J is Jack, it's a non-transgenic control soybean, and H is water. And so here for this particular set of 4 samples, only the after tissue culture one has the band indicating that it is a new NPing insertion site. And we're confirming a bunch of these, and this is kind of a constant process for us. But we, at this point, don't know whether or not this is even a heritable transposon insertion, because transposons love to jump in somatic tissue. But if we're going to use it to study soybean genetics, it's got to be heritable. And so we grow plants out to the next generation. And so we have actually 20 progeny here, so this is the same before and after samples. And so we see these bands are present in the progeny, indicating that it actually, not only did it jump, it was heritable for the next generation, and it's something we can study moving forward. And so we were also interested, well, where are these new insertions located? I showed you that bar chart earlier, tend to be upstream or downstream of genes. And some recent results are suggesting that's still true. This was transformed 9 years ago, so it's kind of interesting to see how its behavior is remaining the same or changing over time. And you can see kind of variable just how far up or downstream, but in that general neighborhood. And it's kind of highlighting some points here. It lands near, but not in genes. But the just upstream landing position kind of begs the question whether or not the structure of chromatin is determining the insertional preference. So for that, we collaborated with Bob Schmitz at UGA using a protocol called ATAC-seq. And I won't go into the nitty-gritty, but it's a genomics protocol that will help define what areas of your genome have closed chromatin, highly bound up with histones, or open chromatin, and generally open chromatins associated with gene expression. And so we have data on this across the soybean genome, and you don't need to worry about the IDs here, but really the right column is the chromatin state, whether or not it's open or closed. And you can see about half of these insertions are in open chromatin. And actually, if you look at the genome-wide average, the genome's probably only 5% open chromatin. So it seems like transposons are preferentially landing in open DNA, which seems intuitive, but it was kind of interesting to quantify. It kind of begs the question, well, if this chromatin was open to begin with, are these NPing insertions affecting gene expression nearby? And so to do that, we followed up on a lot of our insertions. We regrouped plants and collected RNA at various stages. So we have all these candidate genes, and some are supposed to be highly expressed in leaves, others in pods, flowers, roots, really all across the plant life cycle. And so far, all the nearby genes are not affected by the transposon, which I thought was interesting, but I think I have some reasons why that might be. One, we only tested a single tissue for each insertion. So genes are expressed in multiple tissues, and so if we had gone down the list and maybe tested a second or third tissue, maybe we could see maybe some nuanced expression differences. And then the other point is, well, maybe it's not the gene that's right next door to the transposon. It could be a gene further downstream, and that's something with more time we'd be able to look at for sure. And so just wanted to highlight some of the phenotypic diversity we were able to create that we're in progress of studying right now. This is a yellow-leafed soybean, and we had some undergraduates visit from University of South Carolina, Aiken. They wanted to study stomata, so we did nail polish peels, and we think we have some genetic variation on that. And so I've actually been lucky to be able to advise a lot of undergraduates from other universities on summer projects where we go into the field, look at a bunch of mutant soybeans, and figure out interesting traits to measure and see if there are differences. And also, this is really a nice project for outreach. So we have all these mutant soybeans, and a lot of these kids have never been to a farm and get to interact with the plants. We actually have them come into our greenhouse, measure traits, look for differences, see if they can find mutants that we can follow up on in the future, and also take them into the lab and they can actually do some genotyping as well. So it's an interesting instructional tool as well. So some quick conclusions here. So I showed you we have a lineage with active transposition. It seems to be landing in open chromatin, but not affecting gene expression that we can tell so far. And in light of that, we're taking some different approaches moving forward, such as putting in microRNA target sites within NPING and using NPING to move those target sites around to create double-stranded DNA for gene silencing. So if NPING likes to land in promoter sequence, let's leverage that fact and try to cause some gene silencing. And so I'd like to acknowledge our funding source, NSF, and lots of resources from UGA, as well as Nathan Hancock at South Carolina Aiken sent us those summer undergraduate interns, but he also made all the transgenics for this project, and Travis Glenn was very helpful for sequencing advice. Thank you, and I'll take any questions.